In this terrestrial ecocolumn, there was 3 insects, a spider, a cricket, and a praying mantis. The habitat for these insects was very green. There was many sticks, many leaves, and also grass. This is so that the insects would seem like they were at home. In the first week of the experiment, all the animals were coexisting together very well. They were peaceful, they weren't eating each other, and overall they were doing good. The leaves were green and the branches were all still alive.
In the decomposing chamber, there are apples, banana peels, tissue paper, 10 worms, 2 snails, soil, and dead leaves. The worms and snails were the things listed as they were the worms and snails' food. As time went by, these things decomposed, creating nutrients for the snails and worms to strive and live longer. After 2 weeks, the snails died and the worms remained alive throughout the experiment. This was because worms were the top competitor and the snails couldn't compete the worms to get enough food to live. As time went by, we tested the phosphate and nitrates to see if there were changes. At the beginning of the experiment, there was an inadequate amount of each in the soil.
In the aquatic column, the only organisms we had were a fish and an aquatic plant. Yet this was enough to do a good experiment that could compare to their real-world ecosystem.
In the second week, the cricket died. A few weeks later, the spider and praying mantis followed. The grass and sticks were the only things alive in this column for many weeks. After a while, these sticks and leaves died too. There's a second column made for grass which grew, but it soon died after. This was due to many factors, and in general, the terrestrial had no success.
As time went by, we tested the phosphate and nitrate to see if there were changes. At the beginning of the experiment, there was an inadequate amount of each in the soil. Yet, as time went by, the worms did their job and made the soil better, making it a surplus of each of these materials. We measured the nitrate by getting scoops of soil and putting it in a tester mixed with specific dyes that showed whether there was a surplus or deficiency. In the first week, the phosphate was N2 adequate. In week 5, it was N4 surplus. Overall, this ecosystem was very successful in doing its job of providing nutrients to the soil.
In the first few weeks, the water was clear enough to spot the fish wherever it was, but after a few weeks, the water was murky and green to the point where we could not see the fish unless it was close enough to the plastic of the bottle. The fish throughout the experiment didn't die, but stayed alive even though the pH of the water increased throughout the weeks. In the first week, the pH was a 6.5. In the third week, it jumped to a 9.4, and by the 7th week, the pH was a 9.9. The temperature began with a 24.4 in the first week and jumped to a 25.2 in the third week, and by the 7th week, it was at a 25. The dissolved oxygen began at 7 in the first week and was at 8.5 in the third week. It ended by week 7 with a 10.8. The turbidity in the first week was clear and was clear up to the third week. At week 4, it began to appear a little dark. In week 5, it was getting darker. At week 6, it progressed and got even darker. At week 7, the water was a very dark green. Overall, the aquatic ecosystem was successful at keeping the fish alive. This was our ecosystem. Thank you.